Hello, and thank you for joining me. I'm Tori Londrigan, and today I will be talking about the research I did over the summer on the in-field measurement of soil respiration under different cropping intensities. When it comes to agriculture, there are few things more important than the soil. There has therefore always been interest in understanding what characteristics contribute to soil quality. For some time, soil quality has been mostly attributed to physical and chemical properties, such as aggregate size, water infiltration, and nutrient profile. In recent decades, the importance of soil biota to crop productivity has gained significant recognition. Instead of simply a medium in which to grow crops, soil has come to be understood as a complex ecological system with interactions between microbes, macroorganisms, plants, and abiotic factors. Some of the organisms within soil microbial communities include bacteria, fungi such as these mycorrhizae, protozoa, and nematodes, amongst others. One of their most significant benefits to the soil is their ability to convert nutrients into forms available for uptake by plants. Other ecosystem services include water retention and uptake, pest and disease resistance, and many, many more. However, many conventional agricultural practices, such as intensive tilling, monocropping, and pesticide use, tend to reduce microbial communities and their ability to perform these ecosystem services. Luckily, there's been a shift towards agroecological practices that aim to increase the soil's capacity to function as a living system. To know which practices cultivate healthy soils, you need tools to measure them. But the complexity of soil ecosystems can make quantification a difficult task. As a result, most practices tend to target one specific feature of soil communities, such as enzyme activity, biomass, or total organic carbon. One of the most popular methods is the measurement of soil respiration, that is, the amount of carbon dioxide released from the soil. Since many soil biological processes produce carbon dioxide, this method allows us to get a general overview of soil activities as opposed to focusing on one at a time. The two most common methods for measuring soil respiration are pH-based lab methods, in which an indicator changes color with increasing CO2, and in-field gas chambers, which use an infrared gas analyzer to measure CO2 as it rises from the soil. pH-based methods, such as the one on the left, are convenient because you can run many samples at once, as opposed to the gas chamber, which can only evaluate one sample at a time-consuming procedure that also requires expensive equipment. However, pH methods are typically performed in the lab, and lab-based methods almost unavoidably involve the alteration of soil properties. For example, most lab methods involve processing and bringing the soil to a constant temperature and moisture level, qualities which directly affect soil respiration. So, if you're comparing two crop management methods and one naturally retains water better than the other, the subsequent difference in activity might not be detected if evaluated using a lab method. Thus, my supervisor at the USDA was interested in developing a method that combines the advantages of in-field methods with the replicability and affordability of pH methods. And this is what we came up with. Essentially, metal tubes are inserted into the ground at a uniform depth with a pH indicator inside. Once the tube is capped, the CO2 trapped in the chamber reacts with the indicator to turn from pink to yellow. After 24 hours, the intensity of yellow light is measured via spectrophotometer to calculate the rate of respiration. With this method, you can compare the soil activities of different crop management treatments with many replicates at virtually the same time. For our study, we wanted to test the hypothesis that respiration rates between soil treatments will differ for in-field versus in-lab measurement methods. For lab respiration, we used the same pH indicator and incubated the soil for the same amount of time as the field, but the soils were sieved, dried and re-wetted to 50% water holding capacity, and held at 30 degrees Celsius, all standard procedures for other lab respiration methods. The soils we used were from agricultural plots that have been harvested no-till for wheat under varying intensities of crop rotation for the past 7 years at the USDA station outside of Pendleton, pictured above. The three crop treatments are the following: wheat, which is harvested every year, and then wheat, fallow, and fallow wheat are harvested every other year, but are in different phases of crop rotation. And then we also included a native grass pasture to provide a treatment with much different soil communities than the crop treatments. Overall, we expected lab respiration rates would be higher in the lab than in the field due to greater moisture content and aeration, but that differences in activity between treatments would be more pronounced in the field. In addition to field and lab respiration, we also examine extracellular soil enzymes as another metric of soil activity, as well as microbial biomass carbon to characterize the size of the communities examined. To understand the environmental factors that may affect respiration rates in the field, we also took measurements on soil moisture, temperature, and pH. For the sake of time, I won't go into the specific methods used for these factors, but you can find the protocols in the description. Each treatment group had 4 plot replicates for a total of 16 plots. This figure is a to-scale map of the wheat plots used. To choose sampling location, each plot was divided into transects 2 meters in length and randomly selected via number generator. From each selected transect, then 3 samples were collected. The same was done for the pasture treatment, except the randomly selected transects were 3 meter by 3 meter squares. Following the measurement of infield respiration, soil from the tubes were then removed and returned to the lab to perform the other tests. Now we can get to the data. Here we have a comparison of respiration under lab and field conditions, with the different colored bars representing the 4 treatment groups and the y-axis providing respiration rates in units of micrograms carbon dioxide per gram soil per hour. Note that the 2 figures are on the same scale, so rates were overall much higher in the lab than in the field, which we expected to be the case. Surprisingly, while the pasture had the highest activity under lab conditions, it was the least active in the field. We were expecting the pasture to be the most active treatment group under both conditions due to the presence of a diverse array of species year-round, but this was clearly not the case. And then out of the 3 crop treatments, annual wheat had the highest respiration and fallow wheat had the lowest for both methods. For the sake of time, I won't go into the figures for each of the soil characteristics, but I thought it'd be valuable to show you the figure for soil microbial biomass. As you can see on the y-axis, biomass is measured in units of micrograms carbon per gram soil. The crop treatments had more or less the same amount of microbial biomass, but the pasture had much more, nearly double that of the fallow wheat group. To examine how our 3 metrics of soil activity could be related to the other soil characteristics examined, we performed a set of Pearson correlations. The numbers showed our R-squared values, with blue cells indicating a positive correlation and red cells indicating a negative correlation. The more saturated in color, the stronger the correlation is, with asterisks denoting statistically significant relationships. We also ran a set of correlations that excluded the fallow wheat group, as it was the only treatment group that had been without plant cover for over a year. We found the strongest correlation to be between field moisture and respiration, with greater moisture corresponding to higher field respiration. Curiously, we found the opposite relationship for lab respiration. Here we have a closer look at this relationship, with field respiration on the x-axis and water content on the y-axis. I'm going to focus primarily on the blue regression line, as it includes only the treatments with plant cover. We can see that the pasture samples, indicated by gray dots, had much lower field moisture than the crop treatments, which was surprising, as the abundance of established plant and root systems would be expected to promote better water retention, not worse. A possible explanation could be the fact that the pasture is positioned on a slightly south-facing slope, something we hadn't noticed before. Due to this incline, pasture soils likely experience greater direct sunlight and evaporative power, factors that may have been overlooked had respiration only been evaluated in the lab. You may recognize the bar graph above from earlier, and that's because the second relationship worth noting is between microbial biomass and respiration. Whereas more biomass corresponds to greater respiration in the lab, a trend we'd expect to observe for both methods, field respiration is lower with greater biomass. This leads us to believe that lab respiration gives us more information regarding the size, but not necessarily the activity, of a given soil community. Overall, It is clear that lab and field methods result in different measurements of soil respiration, sometimes to a dramatic degree, as with the pasture treatment. The two methods give us different information, both which can be useful depending on the type of study conducted. Suggested by the relationship with microbial biomass, lab respiration provides information regarding potential microbial activity, say after a big rain, whereas field respiration reflects microbial activity in real time in response to environmental factors such as water content. And then we found water content to be the most likely limiting factor for field respiration, at least in late August in northeastern Oregon. As for the next steps, in order for this method to be used in a published paper, it must be validated with an established in-field method of measuring soil respiration, such as the dynamic gas chamber mentioned earlier. And then, to confirm the differences between lab and field, a different lab method that is more commonly used, such as the microplate, should also be included in a following study. We thought it would also be valuable to assess soil respiration rates in the spring instead of late summer, as the springtime is a much more crucial time of year for agriculture. Finally, additional soil characteristics should be included in a following study, such as soil organic matter and nutrient content, to get a more holistic understanding of what's affecting respiration rates, which would hopefully better explain trends in the fallow treatment that we had to exclude from our second regression. Lastly, I would just like to thank the USDA, ARS, and Pendleton for funding this research, and my amazing PI, Kate Reardon, for taking me on this project and giving me the support and guidance needed to grow as a scientist. I'd also like to thank my thesis advisor, Suzanne Alterman, for her organizational and writing expertise, and lastly, my wonderful family for all the support they've given me during my time at Whitman. Thank you very much. And now I'm just going to move to this last slide. So if you want to see the other figures, you can pause and take a look.